Okay, welcome everybody. I can see you coming into the room now. It is my pleasure to be hosting you this afternoon here in Stockholm at least, and wherever you are in the world, welcome. It would be really nice just to test the audio and the technical parts of Zoom while we're getting started, just to give me a quick shout out in the chat or in the Q&A. Let me know where you are from so I can see that you can hear us okay and that you can actually ask us some questions, because we're hoping for some nice engagement today. So while I wait for those to come in, great, I can see a couple of people dropping in there. Fantastic. Hello, hello, hello. It is my pleasure to introduce our speaker today. So my name is Ben Liberton first from SimCell, and we're really, really excited about the topic of this talk. Our invitee, Dr. Kasper Kragh, I hope I said that right, I probably didn't, is assistant professor at the Cost and Biofilm Center at the University of Copenhagen. And he really has specialized in kind of looking into new models, new methods to really fully understand the nature of non-attached biofilm aggregates. And this is exactly what we're going to hear about today. So please welcome Kasper to the virtual stage. As we go along, drop any questions in the Q&A and we can have a nice discussion at the end. But for now, it is my pleasure to hand over to you, Kasper. Welcome.
Thank you so much, Ben, and thank you so much for the invite to SimCell. It's always nice to get to talk about your passions, and this talk will really be about one of my big passions that has really been throughout my career. I've really enjoyed looking into some other aspects of biofilm research than the traditional way of looking at biofilms. And through this talk, I want to talk about these non-attached aggregates. And as Ben said, I also want to fill in some of these model systems that we have tried to develop to capture this specific phenotype. Also, I will go into a bit more depth about some of the metabolic aspects of these aggregates. Now it is a SimCell webinar. I think metabolism should be in this, be part of the talk. Just a moment, I think. There we go. I hope that a lot of you have heard about biofilm before, but if not, of course, I will give a short introduction. And this will also be really a way to say that most of our knowledge about biofilms comes from a guy like this here, a surface attached biofilm, where you have bacteria that is growing on a plastic surface or some innate surface. And we learned a lot from this over the last 40, 50 years of biofilm research. Some of the aspects that we normally attribute to these surface attached biofilms is that we know that they have the bacteria grows these microcolonies and produce this extracellular matrix. This matrix is really a very important part of being a biofilm. And the jump from being a planktonic bacteria that is swimming around to a biofilm, we know that is really very strictly regulated by secondary messengers, so small molecules inside of the bacteria that switches them from the planktonic stage of swimming around to the biofilm and regulates the production of this matrix. One of the reasons why we are really interested in biofilms is that we know they are very resilient towards the immune response. And when we look into chronic infection, we more or less always find biofilms. And one of the reasons why these infections become chronic is because they are not able to be eradicated by the immune response. And the bacteria can actually actively kill the immune cells. Then, of course, we also are very interested in biofilms because bacteria growing in a biofilm are very tolerant towards antibiotics. They can survive 1,000 times the amount of antibiotics as a planktonic cell does. One thing we also know from these surface-attached biofilms is that a biofilm quite often will form gradients within the biofilm, where you have oxygen and nutrients have a hard time penetrating the biofilm and limited diffusion. And this will cause stratified metabolic patterns within the biofilm. So you'll have cells that will grow faster and bacteria that will grow very slow. And this is actually going directly back to the resilience towards antibiotics and the immune response, because slow-growing bacteria will survive more of these attacks. So, but all of this has really been something that we have learned from these surface-attached biofilms. And if we then go in and say, like, okay, but in these chronic infections that we're really interested in, how does they have the bacteria really look when we do microscopy in the infection? And the pattern is more or less always the same, that we see small aggregates of bacteria. All the red here are bacteria that stain specifically for bacteria in cystic fibrosis infection, chronic wounds, COPD. And then we see a lot of immune cells surrounding these aggregates, trying to eradicate it, but they can't. And this is really the norm that we see in most chronic infections. And these aggregates are not that big. Normally, they can be in number in the thousands of cells. And then we often have a huge amount of the immune cells. So the bacteria are floating in an inflammatory response. So you don't have them sitting on the surface. They will instead lie in mucus or pus, surrounded by immune cells. And quite often, this picture before there was a lot of bacteria in that, and that's specifically for cystic fibrosis. You often see a lot of bacteria, but in other infections, such as a diabetic foot ulcers, we actually have a really hard time finding the bacteria. We know that biofilms is there, but it's just very few bacteria that can actually cause the infection. And this here is pictures from courtesy of Klaus Kirketerp and Lasse Quick that has investigated these shavings or debridements from chronic wounds, where they have shaved off parts of the wound to clean it up. And then they tried to really look into where are the bacteria situated in these wounds. And this is, in biologic terms, a really big piece of tissue. And if we zoom in on this small area here, the only place that we can find in this whole piece of tissue is actually zoomed in here and then zoomed in even further here. Then you can see the bacteria lying in small aggregates there. And this also means that it can be really hard to diagnose which bacteria is in there, because if you ran a cotton swab over the surface of this wound, you probably wouldn't find anything. So we also need to adjust our way of diagnosing some of these infections to these small aggregates, because it's not a pus-filled infection. So if we want to say, what is the schematic of a chronic biofilm infection, would quite often be these small aggregates that are floating in this nutrient-rich environment, not attached to the surface at all, and then completely surrounded by these immune cells. So when we think about all our knowledge that we have from a surface-attached biofilms that we have done in flow cells and in microtiter assays, it doesn't really make that much sense to test our compounds, our new drugs, and what we want on those. Actually, we want to do it on these aggregates instead. So one of the things we have studied for many years is really to try to answer, are these aggregates actually biofilm? Because yes, they are in chronic infection, but do they fill up, tick off the whole list that I made before? And one of the models that we have used to investigate this is a very simple model developed by Martin Nielsen and Tim Tolker Nielsen and me and Liang Switty, which is just a agar sandwich where we use simple low viscosity agar, and then cast in bacteria in a small layer, and then the agar itself will restrict the mobilization, and they will grow into aggregates over time. And then we can use a punch biopsy to take out these small plugs that we can then treat in different ways. And then we can see how the effect of different treatments has been. And they form these very small aggregates. And it would take a look, what we used for this was because we wanted to look into the matrix itself. Is that important in a aggregate? Because these very thick biofilms that we're seeing on the surfaces, they can be several hundred meters big. These are not meters, micrometers big. And is it also important to have this matrix, this EPS, extracellular polymer substance surrounding these aggregates? And what we did was we used a lot of mutants where we had either turned on and off some of these very primary components in these aggregates. This is in Pseudomonas aeruginosa, our like main model organism for this stuff. And here we could see that if you changed how much polymers and which polymers that they produced in this model, they gave off these different shapes and sizes. But we could also see that we could pinpoint specific polymers that was really important to survive specific antibiotics. So you had here, in this case here, you can see that alginate, which is really a really important polymer, polysaccharide, that is really important for surviving treatments with meropenem and colistin. Doesn't seem that important with ciprofloxacin. So from this and other studies, we can say that the polysaccharides and the matrix is important for these aggregates as well. We also wanted to look into some of these cyclic GGMP. Cyclic GGMP is one of these biofilm messengers that really moves planktonic cells into being a biofilm. So if you have a high amount of cyclic GGMP inside of your cell, you are really in a biofilm state of mind. I will talk a lot about the bacterial state of mind because I've been working with it for so long, so we have like a relationship. But it's cyclic GGMP in these aggregates. We can actually see that if we add in a reporter into this reporter will produce a GFP if cyclic GGMP is there. So in this plate model, very simple aggregate model, we can see that they light up very, very brightly if they are in the aggregate because we forced them together. So they are really feeling the biofilm in their bones. So we can also say that in these aggregates that we also have these messengers playing a role for the development of these aggregates. One of the reasons why I started being really interested in these aggregates was actually also that we had at some point wanted to look into how much aggregation there are in liquid batch cultures. I think most people who are microbiologists out there have probably seen sometimes aggregates forming in their liquid cultures and maybe not thought that much of it, but we actually investigated this, and what we found was actually the method that you inoculated your liquid culture could produce a lot of aggregation into a batch culture. And actually, if you were a bit lazy and just used a plate colony and put into a liquid batch culture, you could actually get a huge amount of the batch culture being a biofilm state because you had these aggregates floating around. And these were in the size of 100, maybe 50 micrometers across. So you couldn't see them by the naked eye. So you need a microscope to check this up. And what we actually found out was that if you first got introduced aggregates, it would be kind of a snowball effect, and they would take up single cells from the surface. And they used some of these polymers as a glue, and you could actually get around 60 and 70% of an E. coli culture that was aggregates. So you looking at your planktonic culture would just be liquid biofilm, basically. So I don't think that many people are really aware of this. Of course, if you read my paper, you would, but we actually could identify a specific polymer that was gluing this together. So one of the things I also talked about was this resilience towards the immune response. And we have many years ago, Morten Alhed and Maria Alhed, they worked a lot about how these aggregates was not able to be phagocytized by immune cells. And I just wanted to bring this because this is one of my favorite movies that I've ever taken on a microscope. This red part here is a Borrelia aggregate, so Borrelia burgdorferi. And all the green here are human immune cells that are trying to attack the aggregate. And this is in a live-death stained liquid. So if you're dying, you'll produce a red color. So you can see all these happy green immune cells attacking the biofilm and are really continue. And then at some point, then we're starting to have this explosion. And I would love to put sound effects on because it could just be so cool to have them exploding, maybe some fire and stuff. But here you can see the aggregate are actually fighting back against the immune cells and are killing them on going on. And I think this is amazing to see that how much the immune cells are actually being killed by these bacteria. So aggregates are highly protected against phagocytosis and can actually lyse the immune cells. A lot of the work that I've been talking about until now has been in a bit basic media, LB media, minimal media. And one of the model systems that I really enjoy been working with is cystic fibrosis, synthetic cystic fibrosis media that was developed in Marvin Whiteley's lab back in the day. And here we use a media that's really complex and is very, very specific, trying to capture the chemical substances that are in sputum media from patients with cystic fibrosis. And when you inoculate such a media with a bit of bacteria, they will over time grow into these aggregates simply because the viscosity and the high carbon richness of the media. So it's kind of a natural form of the bacteria in these semi-viscous fluids to actually go into a aggregate form. And this media here, you're really trying to mimic the chemical environment. And you can use this media to do live-death treatment and see how antibiotics work. And it will kill the planktonic culture, but it won't kill the aggregates. So it's very closely to what we have seen in other systems. We actually also tried this together with Sophie Dack, where we actually where we added bacteriophages. And there we could actually see that the bacteriophages were able to kill off the planktonic culture and not the aggregates. So that could see that we actually also have some tolerance towards bacteriophages in these aggregates. We actually also added a lot of immune cells to this and saw them attacking the whole stuff. We never got around to publish this, but it was also some good images that we took there. So up until now, I've been talking a lot about these more mechanical and chemical features of these aggregates. And I really want to go with one of my favorite thing about the biofilms is really to understand the metabolism and the physiology of these aggregates. And that really actually was something that started with because we wanted to understand how fast are bacteria growing in these infections. And the reason why we are interested in this, how fast they are growing and the metabolism, is because most of our antibiotics that we use are reliant on the metabolism of the target cells. So, and especially if you have very low metabolism, you are not really that, you can survive quite much higher concentrations of antibiotics in most cases. So if we go back to our flow cell biofilm or our surface attached biofilms, there we know from a lot of studies that the oxygen gradients into the biofilm are really limiting the growth rate in the deeper parts of the biofilm. And it's very well known that there you would kill the surface of the biofilm, but you wouldn't kill the center of the biofilm because it just don't care because it's not growing very fast. And there are drugs like colistin that actually would do the opposite. But colistin is not the best drug in the world, and we try not to use that except in very, very critical cases. Here, these classical images where you can see the biofilm being killed in the center with colistin, and it's killing the surface here with live-death stain. So we are really interested in the metabolism in infections because it can really have something to do with our how effective our treatments is. And what we did all the way back on my master thesis, I think we did this, that we worked on these aggregates in cystic fibrosis patients, because we used a technique where we used these PNA fish probes that are these small peptide probes that will adhere to the ribosomes of the cells, the bacterial cells, but they will adhere so specifically that only one specific probe would attach to one ribosomes. And that makes us actually that we can go in and look at the fluorescent intensity that each cell have based on how many probes are attached inside of the cell. And that will count how many ribosomes it has. So we actually have a neat relationship between how bright you are and how fast you've been growing, how your metabolism. And because this is a wrong way, sorry. Because this is a linear relationship, we can actually go in and look directly, say you are this bright, that means that you have that high of a growth rate. And when we did this in a lot of lung tissue from cystic fibrosis patients, we could actually see that these aggregates, they didn't grow. They grew, but they grew very slowly. It was average around 0.2, 0.1 divisions per hour. So in the infection, these aggregates are just not growing at all, or nearly not at all. And what we actually found was actually that it was actually the immune cells surrounding these aggregates that was controlling their growth, because they were limiting the oxygen that came to the aggregates. So the oxygen had to travel through these activated immune cells, and they take up a lot of oxygen. So there was a gradient towards the surface of the aggregate. So the bacteria is just not growing very fast because they couldn't get oxygen enough. And we actually further down the line, last year we published a paper, me and Mette Kolben and Thomas Bjarnshol, where we actually looked into aggregates in different infections. And there we actually found that the infection, the type of disease that the patient had was actually controlling how fast you're growing. So here, this is cystic fibrosis patients that grew very, very slowly, the bacteria. And this is COPD. And here they grew a bit faster. And then we found in acute pneumonia biofilm, yes, acute infection also have biofilms in them. They grew really much faster. So the state of your infection can actually be attributed to how your metabolic activity is. And that could maybe also explain something about if the treatment is working or not working in these infections. So again, we have a very stratified growth system. Bacteria can grow faster some places within the same infection. It can be different parts of the infection that can have slower, and also within the aggregate itself. And this leads me into just a good example of how you can also mimic this. This was done by Amy Wessel in Marvin White's lab a couple of years ago, where they used these small houses for bacteria, these microtraps, where they then could capture a bacteria inside of these small houses, and they will grow over time into these aggregates that was on these stills. And these houses were permeable for media, so they could flow over the surface of the aggregates. And then they could see over time that anaerobic, they used these GFP reporters that would turn on GFP if they came into an anaerobic condition. So they could see that the center part of these aggregates was actually becoming, started to light up. So you had gradients inside of the aggregate itself. So besides what we have in the infection, there's no immune response or anything here, but the aggregate itself actually also have a gradient within them. So it's really complicated the way this stuff grows. So we have the same gradients as we talked about before. Another way of studying this that Maiken Sønderholm and Mads Lichtenberg developed this model where we try to capture both the external gradients towards the aggregate, but also the internal gradients within these aggregates. What's this aggregate alginate bead model, where you produce these small beads where you capture bacteria inside of them, and then the bacteria will grow into small aggregates that will be more or less the same size and conditions as you have in cystic fibrosis. And this is just a really neat small system where you make this alginate mix, and then you drop it into calcium chloride, and then they will like harden, solidify very fast in these small beads. And then you can grow them up to bacteria, and you can throw them into different kinds of treatments. You can throw them into the calScreener. You can use all different kinds of treatments on them. And what we found in these systems was that we could see that the bacteria at the outside grew to much bigger aggregates than they did on the inside of these beads. And by measuring their growth rate, we see that the outer part grew much faster than the central part, which makes sense because they were big, smaller and bigger. And we could also measure that there was these steep gradients towards the center of these alginate beads. So here we try to create these external gradients towards the center of these beads. And if we treated these with antibiotics, they could survive 100 and nearly 1,000 times the minimal inhibitory concentration of most of these drugs. So they were very tolerant, these bacteria in these beads. So me and Mads Lichtenberg has been trying to be working on some kind of models to visualize like this, all these gradients and different growth rates. Like, how do we think it is in these aggregates? And a free floating aggregate, an aggregate that's just floating in a liquid, would have this gradients towards them because they're taking up the media cell. But most of their gradients would be internally inside of the aggregates. So you have cells that will grow slower and slower in the middle and then faster on the outside. But in some of these gel-based systems, here you have a diffusion gradient where you have a rich upper part, and then you'll have diffusion and gradients forming towards the aggregates and below the aggregate itself. So here you get a much more complex system. And then here compared to what we know is happening in the surface attached biofilms. But then let's try to put this into the very complex system of a chronic infection, because then we can have bacteria positioned somewhere in this is a drawing of a chronic wound, because that was the best example we could come up with. But here you have a oxygen coming in from the surface, some bacteria growing slowly, some bacteria growing fast, the immune cells surrounding it would, and we just end up with a really, really complex system that is just very hard to understand. And this is why we really have to learn much more about the metabolism of these biofilm aggregates for us to understand simply what are we looking at, because it is such a complex system. So up until now, I've been talking about growth and I've been talking about metabolism, more or less like that. This is the same, but our latest study have showed us, no, it's not. So we wanted to understand how this metabolism actually works in this. And there we used SymCell's calScreener system. And I think probably a lot of you have probably heard about it before, but I will give a quick rundown. The system has these small plastic inserts that you can throw into these titanium vials. And when these vials are then sealed, you can throw whatever bacteria you want into them. Then whatever heat or energy that is being produced inside of these containers will be very precisely measured by the calScreener. And the metabolism of the bacteria inside of it, or human cells or whatever you put in, will be registered as this spectrum of this pattern of metabolism that are in real time registering how much is happening right now inside of this vial. And these patterns will be quite unique to each bacteria and types of species of bacteria and what carbon salt they're growing on, and also which state they're in biofilm or not biofilm state. And that was what we really want to capture with this system. We want to see if we could find a metabolic fingerprint for biofilm. So we set up a system where we grew some surface attached biofilms inside of these inserts for the calScreener, and we looked into some planktonic cells, and then we used this alginate bead model as a non-attached aggregate model. And then we over time got these neat metabolic patterns. And with the use of some principal components and analysis and some machine learning, we were actually able to identify each one of these systems how they grow. They cluster together, and we trained a machine learning algorithm that is actually able to, if we give it a blind sample, it's able to say that it's a surface-attached biofilm, a planktonic culture, or a surface non-attached aggregates. So that was really neat for us, but we wanted to know more about. But yeah, that's the metabolism. But it's what does it do with this metabolism? And here we came back to our cyclic di-GMP messenger, the biofilm messenger before, and we produced strains that was producing a much, I was about to say some bad words, lot of cyclic GGP, and then we produced strains that didn't produce it at all. And we could see that the spectrum in this model, especially here in this alginate bead model, here we were forcing the bacteria together to be a aggregate. And there we could actually see that it was completely different if they used a lot of this or didn't use a lot of this cyclic GGP. And normally it's never been reported that cyclic GGP had anything to do with metabolism or growth, because people have been looking into growth, not metabolism. So there was no people saying that doesn't control anything with this. But yeah, it's not important. But what we found out was actually that if we took these bacteria that was super high in cyclic GGP, and they were really in a state of, I'm a super biofilm producer, and then removed all their matrix components, so it was not able to produce any matrix at all. Then here, this yellow, that's a super producer that's spraying out this matrix components. And then we have here the normal wild type bacteria. And then down here, this is a super producer that's just not producing anything. And the difference between these two is all the matrix components. And this really gave us the understanding that when you're in biofilm, yeah, you might not be growing very fast, you're not dividing very fast, but you're using a lot of energy to be a biofilm. It's really expensive in energy to be a biofilm, but it might be worth it because you're protected against all this nasty stuff we're throwing at them. So, yeah, the last part of my talk here will be a bit different, because this is actually something I did together with Syncell and on a big project from Horizon 2020 a couple of years ago with universities from all over Europe, and we were a big consortium of labs. And here we were working on using this system, this calScreener setup, to screen for combination treatments, synergistic treatments, where you could actually put together two antibiotics and then get a super potent treatment. And what we did was that we were, all these many labs all over Europe, they screened a lot of multi-drug resistant pathogens that was super resistant for a lot of different types of antibiotics. And in the calScreener, they would use that to mixing some of these drugs that they were actually resistant for in different combinations to see if we could predict some effective synergistic treatments. We found a lot of these, and then around 13 of these strains, we took these combinations and challenged them in a mouse model where I used that was a lot of killed mice. It was horrible, but we got some fantastic results. We actually found out that the calScreener's prediction could very precisely predict if a combination treatment would be effective in this mouse model. So in theory, we could, in the very long run, maybe we could use this as a way to predict and say, okay, we got this strain in from the lab. It's super multi-resistant. What if we test all these different combinations, and then we can try to give patients a new combination that has synergistic effect, so you can actually get a treatment that works. And this is not done in the calScreener, but a paper we just got accepted, done by Eva. Oh, I'm going to butcher this. Sorry, Eva, you're listening. Sorry, from Barcelona. And me and Tim Tolkien Nielsen and Niels Højby and a lot of other Chufu. Here we investigated these highly resistant, colistin resistant isolates from cystic fibrosis patients. These strains just didn't care at all about the colistin. And when we grew them in a biofilm and added a rifampicin, we could move the biofilm hereditary concentration, so the amount that it took to kill the biofilm, from around 250 micrograms per milliliter to 2. This is one of the most potent combination treatment I have ever seen. So by adding rifampicin, that doesn't really do anything on its own, but it opens up the bacteria for this. And this is, I've been really wild with this. And we tried then in this surface attached biofilm assay. And as you see here, like colistin on its own doesn't kill anything. Rifampicin on its own doesn't kill anything. Combined, they eradicate the biofilm. And this really made me think, and this is just an idea I have. I want to put it out there. If anybody want to run with it, it would be fantastic. Because what if we could combine biocalorimetry and in the calScreener with this knowledge? How about if we took patient samples from a cystic fibrosis patient and grew bacteria in these inserts in a agar gel as aggregates? And then we could use the calScreener to scan for combination treatments that would be potent towards this specific strain. This would, in theory, if it worked, it could give us a personalized medicine treatment, a regime that would be specific for this patient, that could, and by using the calScreener, you could get around biostatic effects. We could see that only bacteria that are actually being killed in a biofilm here. I think this could actually be, if this could work, we could really make a fantastic diagnostic tool to treat cystic fibrosis patients. So with that, of course, this is 10 years of work. So that's a lot of people that has been contributing, and I hope that I haven't forgot anybody, but especially Tim Tolke Nielsen and Thomas Bjarnsholt has been some of my mentors all this work, all the years, and a lot of funding agencies have thrown a lot of money my way. And of course, Symcel has not thrown money at me, but they have helped a lot with technical support. And then lastly, I just want to say I'm going to ECMID in Copenhagen and ASM in Houston. So if you want to talk, I always would like a cup of coffee and discuss biofilms, aggregates, calScreener, the techniques. And with that, I will send it back to Ben.
Thank you very much indeed, Kasper. That was extremely inspiring. Just before some of you have to run, I know you have to dash. Thank you very much indeed for attending. And please, if you have any questions, you can just respond to the email that you got with this webinar, and we'll be happy to follow up. But in the meantime, please, anyone who's staying, go ahead and ask your questions here. We've got time for a discussion. I'll start by expressing my my gratitude there, Kasper, because I was getting a little bit depressed, kind of like, oh my goodness, what are we going to do in the clinic? You know, none of the biofilm models are actually representing the real situation. How can we actually come to something that can actually make a difference in patients? But yeah, you came to it in the end, and that's super interesting to see. I mean, that must be kind of a relief as well, I guess.
That we hope so. That could really be I think that has potential to really make a difference, and also especially when you tailor it specifically to the patient. I think that is the key. That there is no silver bullet in when it comes to biofilms.
Cool. Okay, I've got some questions coming through now. So if I've got two questions relating to multi-species biofilm. One is like, in the aggregates you had, did you have other species? Did you try with things like Candida, for example? And the follow-up question is relating to how would that look in the calScreener? You know, do multi-species biofilms look different when you look at the thermograms and the heat traces?
Yeah, first of all, we have tried to do combination, and we've done that in some, especially also some other wound models. We have a wound model called a semi-solid model, where there is really important in wounds if you have a multi-species infection. Of course, I would always say that one of the things, it's very rare, if ever, that we actually see a multi-species biofilm where you have them really combined, but we do see them in the infection together, and that would often give a much more potent infections and much more violent reaction. And yeah, we have done that. We also tried to candidate actually in this alginate bead model. That was actually really cool to see that we actually could see that candida went into a hyphen state very fast in this model, which can sometimes be indication of a kind of protection effect, because candida can both be in a yeast state and in a hyphen state. And that was kind of interesting for us, because there we had Staph aureus in the bead itself, and we actually see that the candida laid at the outer edge of the bead in this hyphen state. And also generally when we're doing diagnostics, we don't want to have a hyphen state in a patient, because that normally is a quite terminal indication that it becomes very violent at that state. But yeah, we have very limited tried with multi-species biofilm in calScreener. I know there's a lot of other people that has done that, and it can give a very the combination of the spectra will be very different than the single species biofilm. And I think that could be really interesting to try to see in a model like that, if you can actually alter the metabolic pattern, if you have them closely related or further apart in a immobilized state in the calScreener, because that would be a more relevant clinically that we have them in that way. But I know that people are working on extracting these metabolic patterns, so we can actually see multi-species and identify that in directly. So I think that would be a lot of fantastic work that we could do, especially with the calScreener that way, because we could identify maybe faster, because when you're doing a plating, you only see what come up on the plate, and it could be that you're only getting the species that is most prevalent of grows faster.
Great, thank you very much. I have a follow-up question, I think, related to that. It's can the microcalorimetry setup be modified to monitor phenotypes of different cellular populations? So I guess I wonder if that's not just referring to multi-species biofilms, but potentially also the kind of examples you showed with human immune cells as well. And specifically, like, how do you set up the calScreener to do that? Is it straightforward, complex? What do you need to consider?
Yeah, I know we're working on it, especially with the immune cells. That is really something that we have been really interested, especially Thomas Bjarnsholt's group. They were working a lot on looking into especially some of these innate immune response cells. That could we see if they become activated? Can we see a different state there? Can we see if the bacteria is there not there? This interaction between the immune cells and the bacteria, can we actually measure that? Also, could we put that in some case in the end into a both diagnostic, but also a more if we took samples from patient, could we see a higher metabolic rate because you had a lot of inflammation going on? So using that as a biomarker. I have not worked that much with it, but I know that they are working a lot with that. And I think there's a lot of possibilities. And basically what you would do is that you need some baseline measurements, a lot of measurements that you can know how much is the unactivated, not the state. And I could imagine that you could also probably use it for cancer cells, HeLa cells, all this stuff. See if we can get any expression that is different when you activate and not activating, use hormones on them, all these kind of things. That
cool. Yeah, There's a world of possibilities. I have another question here about basically what is the difference that you have seen in your work in the antimicrobial resistance profiles of the non-biofilm aggregates compared to normal biofilms in the CF model?
In generally, or
yeah, I think so. Maybe the difference in the resistance, in an aggregate compared to non-aggregate.
Yeah, but we actually haven't seen that much of a difference, and that's probably maybe a bit when you say it like this, there's maybe not that much of a difference, but that's also really one of the important parts of, I think, our research is to get these aggregates recognized as a completely real biofilm, and there will be the difference that you have both diffusion limitations in that a lot of the polymers that we have in a biofilm will we know that they can take up some antibiotics if they are negatively charged, if they're positively charged, and so the matrix itself, if you have only one front of attack as you have in a surface attached biofilm, that would be different than if you have aggregate floating around. But so there's a lot of these very basic things about surface to volume ratio of how much can you get your immune antibiotics. But right now I haven't seen an aggregate that I could eradicate very easily. So I think it's more or less the same.
Interesting. And so you talked a lot about the kind of personalized medicine approach where you'll be like really screening the aggregates and different antimicrobial combinations. But did you get any trends or patterns where you could say certain combinations of antibiotics normally kill the whole biofilm aggregate, or is it not as simple as that?
What I've seen up until now is that you need to really combine the treatments in a way where it gives a edge for the other antibiotic, and what we believe in with we know that with meropenem and colistin, they are very potent together, and it seems also that avibactam and meropenem has some activity together, and quite often it is that you either overwhelm some resistant parameter or that you're actually opening up. We think that with this that I talked about with colistin and rifampicin, that rifampicin is actually opening up the access for the colistin to get in and attack its target. So quite often there will be some mechanisms that make sense that you have but it could also be very simply that you have that you're just targeting two very different subpopulations, as you do with colistin and tobramycin or ciprofloxacin. There you will attack both, and that's why we use that for cystic fibrosis patients today as a treatment option, because we are trying to kill both subpopulations.
Yeah. Interesting. I have a specific question here about antibiotics. So combinations of colistin with IV fosfomycin for eradication of Acinetobacter or Pseudomonas. Do you have any insight into that from your work or any experience?
No, not specifically. No. That's definitely also, like you say, IV, because that's definitely also the delivering methods. It's also really important in all of this. Like how do we get it into the patient? Do we use it, give it in IV or peroral or inhalation or whatever? That's something we need to take into account even further.
Yeah, complicated. So when you take samples from patients, do you see biofilms growing on kind of patient molecules? If you're extracting like fibrinogen or plasminogen, would you get biofilms attaching to those kind of matrices?
I would think. I haven't personally looked into this. I know that there's a group in Seattle that has been working on some of these macro molecules in the sputum of the cells that through some different mechanisms that they will create a osmotic gradient that will push the cells together in aggregates. But I haven't looked into specifically on growing on the surface of these.
Yeah, it's crazy. These questions, just kind of really highlighting how complex the real situation, in a patient in the hospital is. And it's why we need these models, I guess, isn't it, to kind of really try and simulate that in some way, some meaningful way in the lab.
Definitely. That's definitely, yeah. Go ahead.
So how do we make sure that the aggregates that you see are not just dispersion from a surface attached biofilm?
That's of course of course in infection it would rarely be that, because we don't have movement in that way and it's really been that, like, I've been looking through quite a lot of infection and a lot of my colleagues has been, and I think we have seen biofilms on the surface in the intestines and in the teeth, and that's about it, where we have seen a real surface attached biofilm. So we just don't see where they come from. But I think it's really important in other model system, the attachment upstream aggregates, that you will have dispersal of aggregates, and I might think that it's even more important with aggregate dispersion than planktonic cell dispersion. But yeah, it's in the infections, I don't think, because there we rarely would have any attachment to the surface anywhere, and especially in some of these, it's very viscous environment, so you don't really get that much flow in any way. But yeah, in many other settings, in industrial settings, in environmental settings, I think that these aggregates are part of the dispersal strategy from the bacterial part.
Yeah, for sure. So it talks about inoculating your cultures with a colony, and actually, we've gone from kind of the normal biofilm model saying you're not studying biofilm, that's not real biofilm, to actually when you do a normal planktonic culture, maybe you are studying biofilm there. The question here we have is, like, does the age of the colony actually affect, the aggregates that you'll get in a culture? Like, have you seen anything there?
Actually haven't looked into that. That would actually been fun to see a older plate, like if that changed things. These was just overnight cultures overnight plates that we used, but of course it would change. There's so many things that would change with age of a colony. That's also basically a colony on a plate. That is a biofilm as well. Let's just be honest. So it's that would definitely be fun. One thing I also said that we tested these cultures on their, like, the whole culture, how their tolerance towards antibiotics was, and that was significantly changed in the ones that had a lot of aggregates. So if we just added antibiotics, most of the population would survive when it was mostly aggregates. So, yeah.
Wow. What do we know about the EPS, Kasper, of the aggregates compared to a surface attached biofilm? Is there any difference there in the composition or how it's expressed?
We haven't found any difference yet. The one thing that we have noticed is that specifically, what we can do, we can both remove a EPS factor, we can remove PEL or PSL, but we can also overexpress it to see if we can add on to it. And if we overexpress a aggregate biofilm, there we would actually see that especially with some of these PSL and alginate, that it becomes a bit fluffy. There's a bit further apart between the cells, but as they are still immobilized in a bit, they will still be there. And in a surface attached biofilm, especially if you have some liquid dynamics going on the surface, quite often they would actually be released because it's not as strict. The biofilm doesn't have the same rigidness in it. So there we can see also, if we remove PEL or PSL, then we will get a less amount of biofilm in flow cell biofilm, but we will still get the same size of aggregates, more or less. So that's different, but I think that will be a lot of possibility to study that. So we just haven't gone around to it yet.
Cool. So we have a very important question here as well, Kasper. So what on earth do we do if we want a clonal population of bacteria? So they're kind of saying, if one cell in liquid broth, the normal kind of protocol is actually causing aggregates. Do you have a recommendation as the way to actually do it, or is it just impossible? We're never really studying clonal populations.
We actually, when we wanted to investigate something that is really planktonic, like for example, looking in our calScreener wells, there we didn't want any aggregates because we wanted to study the planktonic culture, and there we actually used these large size filters that we had with the pores that was 10 micrometers in the diameter, and then filter the culture through it. So we remove the aggregates and get the planktonic culture. Of course, then there's also the possibility that you're selecting for cells that are not in a biofilm state, but that's really been the way that we have done it. When we it's really important for that. There's also a small trick that I learned over the years. It's like if you have a small overnight glass tube culture, that if you leave it on the table for 10 minutes, most aggregates would actually fall to the bottom. So if you take in the middle part, then you get rid of a lot of them. But yeah, but I don't think any clonal population doesn't exist even in a liquid culture, because you will have mutation all the time anyways. But yeah, that's it.
Oh dear. But yeah. No, that's super interesting. Got another question here. So have you tried combining the work of Mette Kolpen with the hyperbaric treatments with your work on the prediction for the personalized kind of antibiotic treatments?
Nope, not yet. That would be that could actually be really fun. Because that is definitely both her hyperbaric work, but also her norm-baric work that is actually using just pure oxygen. And for people who don't know that Mette use a pure oxygen mix to get rid of these gradients that we have around the bacteria. So simply getting them up in metabolic activity by like squeezing in oxygen. But that would be super interesting. Also, just to see how the effectiveness in a calScreener or anything would be. That could you give it a huge go with the pure oxygen and see if we can actually maximize the metabolic activity and the killing from these. But yeah.
Great. And final question we have here relating to antibiotic resistance potentially causing even more problems for treatment of biofilms in CF. Is there anything to think about for the future? Are the models and systems that you develop kind of indicating a way forward to try and treat CF biofilms in a different way?
Yeah, but the thing is that right now we started to become really good at keeping CF patients alive for a very long time, but it also means that they have to be treated with huge amounts of antibiotic over a very, very long time. I think there is a talk about that changing the regime of the treatment, which drugs you're using that are targeting different, because a CF patient will get like kilos of antibiotics over their lifetime. So I've heard people talk about that they had to switch between some of these, so we don't create these populations of hyper resistant strains, because it is just a really bad combination of treating the same strains again and again and again with this huge amount of antibiotics. So yeah, changing the regime, adding stuff like Mette's hyperbaric treatment options, because there you're adding in different dispersal agents. People are working a lot on that as well. I know there's also people who are working with various phase treatment and different combinations of these could really be important. Also, that's just from the top of my head, that if you select it without having treatment on it, but select it for fast growing cells by adding oxygen treatment or something that you can actually select, because most of the bacteria that will be hyper or very highly resistant will grow slow, because there's a cost to being resistant. So if you select it for fast growing cells, that might dilute some of this resistance. But no, I don't have any clever answer. That's
well, thank you very, very much indeed, Kasper. Thank you to everyone who has attended and for all the great questions. We've really kind of kept us going right up until the hour, which I appreciate. The recording will be available and you'll get access to that as soon as it's ready. So yeah, thank you very, very much indeed, Kasper. It's been a pleasure to hear about what you've been doing. So thank you.
Thank you very much for the opportunity.
No problem. Speak to you soon. Bye-bye now.